Now it's a great pleasure that we come to our keynote speaker. It is Professor Paul Herrling. Allow me just to say a few words to him. He is the chairman of the board of the Novartis Institute for Tropical Diseases in Singapore. And he is also professor of drug discovery sciences at the University of Basel. Before his official retirement, he held various positions in Novartis. He was, for instance, head of corporate research, and he held also many other jobs. So it's a great pleasure, Professor Herrling, to have you here. He will talk to us about the next generation malaria medicines.
Thank you very much, Professor Boone. Thank you for the invitation. I don't usually speak about nanotechnology or nanomedicine, but on the other hand, if you think as a pharmacologist, you know that most of the drugs you work with are very small. So you could say, you could include it in nanotechnology. But of course, why there might be an interest for the public at this meeting is, of course, what I've seen, and the next talks will address that. Very often, getting the drugs to the right places in the body is a problem. And that is where I see that nanotechnologies might be of great use. So what I'd like to relate to you is the story how we're getting to the next generation of antimalarials. Malaria still kills hundreds of thousands of people per year. So it's not a solved problem. It's not very close to elimination yet. There are a few pockets, like in Tanzania, close to where the Tropical Institute did studies where the case numbers go down, but it's still hundreds of thousands. And you can see it's mostly a sub-Saharan African problem. Even so, Southeast Asia has the problem too. This is the culprit, so that's the parasite. I will speak today only about the falciparum, Plasmodium falciparum. I will not address Vivax because we don't have time, and so we'll work on that too. And the vector, as you all know, is a mosquito. And we've seen this picture in Dr. Bruhn's talk just before. The symptoms occur when the parasite goes into the red blood cells and eventually destroys them. It is one of those biological organism that has an eminently complex life cycle. I will come to that a little bit later. You will see that even its pharmacology and genetics must be extremely complex. Anyway, I'll go very quickly here. Let's start the cycle here. The gametes are in the gut of a mosquito that has taken a blood meal from a patient that is infected. They evolve into different stages, and the oocyst then moves on, goes to the salivary gland, and there are sporocytes that are produced. And the salivary gland is important, of course, because saliva is being injected whenever the mosquito picks a patient so that the blood doesn't coagulate. And at the same time, they give us a gift of sporocytes. Those go straight into the liver. By that time, you have no symptoms. And they evolve further, and then they come out of the liver as merozoites, go into the blood cells. And there again, there are several stages. And during this time, when they start to kill the red blood cells, is when you have the symptoms, as I mentioned. And eventually, sporocytes are again produced, the gametocytes here. And these are the ones that then continue the cycle at the next blood meal. Again, the Vivax is a special case. It has a dormant stage in the liver, but today I won't be able to talk about it. Why do we need new compounds? Even because you all know that we've had antimalarial since in Europe since 1600. Actually, the Incas had the treatment of quinine much before that. So the 1600 is the year where the Europeans became aware of that and brought it to Europe. What I want to show here, the cross is, the point is when one of these antimalarials was discovered in Europe, and the cross is when resistances occur. You can see in quinine it took nearly 300 years, and the reason why is because it was only sporadically used during all this time. But when it started to become used in a massive way, mostly during colonization, and mostly not for local patients, but for the armies that were going to these places, that is when large populations were exposed to any one of these anti-malarias. Then, of course, you put a selective pressure on the population and you get escape mutants and resistance. So you see the ones that came afterwards, they all eventually, within a few years, and the most extreme cases is here Proguanil that showed resistance in the same year that it was used. The current treatment, the best treatment today, the standard, is a very old, there is a derivative from a very old traditional medicines from China, that's more than 2,000-year-olds, is Artemisia annua, is a plant, it's nearly a weed, and you can extract a compound from there that is extremely potent as an antimalarial. And a collaboration between the Chinese scientists and Novartis actually at that time led to the current standard of therapy, which is a combination therapy of artemether and lumefantrine. It's always better to treat a parasite with two agents at the same time rather than one. That reduces the probability of resistance. However, what's happening, there's always a few clever criminals who, despite all recommendations, use these things as monotherapy. And that happens also with the artemisins, and this led then to the emergence of resistance, certainly in Southeast Asia, in the Cambodia, Thailand region. And about 7 years ago, the Wellcome Trust came to us because we had founded this institute in Singapore to work on neglected diseases, and said, these are the first signs of resistance. We need to work now on the next generation. And that's the story I will show you in the next few slides. So what we wanted is a one-dose cure. I mean, today the standard of therapy is 3 days with 4 pills in the morning, 4 in the evening. And the practical problem of that is simply that people in the endemic regions have a very bad supply system. They never know if they will get the next dose or not. And endemic regions also mean that people get reinfected all the time, and it's very often it's children. So when the mothers have a pack of Coartem for 3 days, when they see the patients or the children improve on the second day, they keep the third day, just in case a reinfection occurs. And of course, you know, by not finishing the treatment, they select for resistance. So it would be great to have a one-dose cure. I will not address this, as I mentioned, so it has to be very efficacious. It has to be a new mechanism of action to overcome all known resistances. It has to be safe because you use in pregnant women and young children, orally in 3 daily doses or less, and that is ideally 1. It has to be very inexpensive because that's a region where no patients have money. And it has to be stable because it's in a tropical environment. You cannot afford a cooling chain. That's the place where this happens. It's about 100 scientists there, but I would like to point out these 100 scientists don't work in isolation, but they are part of the entire Novartis research network, which is shown here, about 6,000 scientists. And I can also say, having established this non-for-profit institution in Singapore, has created a huge motivation with all our scientists who otherwise work in the commercial area, and they have been outstanding in helping out whichever way they could to complement the technologies we didn't have in Singapore for these topics. We work there on dengue, malaria, and sleeping sickness. So we started, as was mentioned before, about the library. So we have a big libraries of compounds, and the first thing that you do is you try to see if any of the compounds you have there contains a hit, is the compound that might kill Plasmodium. We have an institute in California that's specialized in engineering and biology, and they have robots that can screen millions of compounds in short time. So I won't go to the detail of that, but starting from 2 million compounds, we got 4,800 hits. That is all compounds that somehow killed the Plasmodium. I would like to say a very key issue here is that we had an assay that reflects the biological situation in patients. And that is human blood infected with Plasmodium, with a parasite that has been genetically engineered to be fluorescent. So if you have a drug that kills the parasite, the light goes away. And this way you can test millions of compounds, and as I said, in a natural environment, because that's where the Plasmodium lives, at least one of the cycles, is in the blood cells. So what you see in that assay has a high likelihood to be predictable for the whole, for the patient. So we got 4,800 here. For those then we selected for higher potency. We got 1,200, and eventually 200 scaffolds, so different chemical classes that had this property of killing the parasite. By the way, we call this the malaria box, and it's available to any scientist on the planet. So that's not, we haven't patented in the sense to make it proprietary. It is available actually through the Medicines for Malaria venture that you heard about before, one of these non-for-profit organizations. They have access to these. You do dose response curves. We selected the most promising scaffold. Then you do the medicinal chemistry, which I don't have time to discuss here. The compound we then selected was this one. For the experts here, you can see it has some similarity to a natural compound by having several asymmetric centers. But it's a small molecule. And it was extremely potent, and it looked rather safe. It was extremely specific, and you will see how specific it is afterwards when I show you some of the results. Interestingly, it had never been used anywhere in medicine, and which indicates the specificity. It had never been seen in any other disease or screen, so it made it probably safe with very little off-target side effects. It was synthesized in 2007 and declared a lead, that is a compound that might make it to the pipeline. The first stage, that's a preclinical candidate, was in 2008. And then it was ready for the clinic in 2010, and we're now in phase 3, as I will mention a little bit more details before. As we thought, and here look at this one, we compared it to several of the known antimalarials in an animal model. That is Plasmodium berger in mouse. In this case, it's slightly different, the different parasite that lives in mouse, but it is predictable to man. And you can see it was, in this assay, it was at least as potent, that's 6 to 9 here, this is this new compound, as potent as the attenuates. And certainly more than chloroquine and these older compounds. Now here, I'd like to spend just 1 minute, a little bit longer on this one. So what we did up to here is what we called phenotypic screening, which means we don't go for a known protein in the parasites and we declare that as a target, and then we find compound that affect this target. What you do is you take the whole organism, you take whatever kills it, and you worry later how it does that. So at that time, we then thought, okay, it looked so interesting, we now also would like to know what is the target, what is the molecular target in the parasite that actually kills the parasite. And this you could do by using a method that was not possible a few years ago. And that was cheap, deep sequencing. So what you do something you don't want to happen in nature. You take a culture, as I mentioned, blood infected with parasites, and you expose it now to a sublethal dose of this new drug. And after a certain time, you harvest the survivors, and you do it again, and you do it again, and you do it again. After dozens or hundreds passages like this, you will obtain parasites that are completely resistant to your new drug. And then you do the thing that you couldn't do before. You take these parasites, you compare them, the wild type where you started with, and you compare it now to the resistant form, and you sequence the entire genomes from A to Z, the whole thing. and you see if you can find differences. In this case, we could. We found different in a gene, which was a gene for Plasmodium falciparum ATP4. So it's an ATPase. Now, a pharmacologist, if you have told him to go and take an ATPase as a target, he would say, no way, ATPases are all over the place. It's unspecific. It's never, you're not going to have terrible side effects. By not knowing, we picked it out, and by that time we knew it was rather specific, and I'll show you in a second how specific. So all the mutations we found in the resistance were all in this protein coded by this PF-ATPase4 gene. So the likelihood that that is the target is pretty high. So now we ask the question, what does this ATPase do? So what it does, long story short, it regulates a pump for sodium. The biologists and physiologists amongst you know that sodium is a key ion for regulation of the membrane potential of every cell in the body. And if this membrane potential collapses, the cell dies. And so this and there is a lot more sodium outside the cell than inside. So in order to survive, cells need to take the leakage of sodium into the cell and pump it out. And that's an energy consumption, consuming process. This is why it takes this ATPase. And it seems that is the ATPase that does that in this Plasmodium falciparum. Okay? We'll come back to this thing a little bit later. Now, so by that time, we had done all the preclinical safety. It was a very clean compound. We went to Australia. We did the so-called phase 1, where you test it on volunteer students to see if there is any sort of unforeseen effects that you had not seen in all the previous testing. And that came out clean. We could get pretty high doses, which was much higher than the dose that we predicted to be effective then later in patients. But you need to do that to see how you can go until you can get some side effects that might be limited. Then, when this was all positive, this so-called proof of concept in human volunteer, we had to go in regions where patients are to test the efficacy. That's what we call the proof of concept in this case, in man. Namely, does this drug actually clear parasites in people? And these were 3 sites in Thailand where, actually in regions where the new resistance to the artemisins are emerging. But we did not select specifically artemisinin-resistant patients, because there weren't that many at that time when we did that. And the result was absolutely astonishing. You can see it here. I'll give you just one result. What you see here is a number of patients that were tested. These are quite a lot of antimalarials known. And here we use one measure to see the efficacy of this thing. And it's called the parasite clearance time. Essentially, how many hours does it take after the beginning of the treatment until all the parasites have disappeared from the body? And you can see that most of the older antimalarials are around 100, 65, 70 hours, so several days. The artemisins are 50, so that's 2 days and more, about 2 days for the artesunate. And this new compound, 12 hours. It is the fastest parasite clearing compound ever seen in history of medicine, which gives us hope that we may be able to get very much shorter treatment duration, as I had in the postulate at the beginning. We are now in the process to actually see if we can go with the dose high enough to do a 1-dose cure. And 2, the second big piece of work we need to do, we don't want to give this compound alone. We need to find a partner to combine, as I mentioned before, to prevent resistance. Now, I will just summarize, as we have other compounds. Meanwhile, as I said, I couldn't talk to all of them, but here is again the life cycle of the parasite from the liver into the blood, and into the blood you have these stages that kill the blood cells, and then the gametocytes that are being resucked up. So this compound that we have, that I described to you now, is specific for these blood stages. It can also kill the gametocytes, which is good because it also reduces or stops transmission. Now, what is totally surprising when we think of the target, so this ATPase, a fundamental enzyme to keep the membrane potential alive, well, this works only in these blood stages. It does not work, it doesn't kill the liver stages, meaning that these subtypes are the genes that are important for survival of the stage are different at different stages. So it's not only specific for Plasmodium falciparum, this compound and this target, but only for one piece of its life cycle. Very interesting. We do not have expected that. But meanwhile, we have other compounds that can affect the liver stages. All right. So this was, as was hinted, a huge collaboration with a lot of people. And you can see that the Swiss Tropical and Public Health Institute here was prominent and contributed to this project. I mentioned the group in California, all the other colleagues within Novartis, but we were also the funders where the Wellcome Trust, very importantly, and MMV, and through MMV, the Gates Foundation, and of course Novartis. And of course, I didn't do this job. These are the people in Singapore who are key in discovering and developing this compound. Thank you very much for your attention. Not bad.